this is part 2, looking at the life cycle of a modeling project, in particular, modeling the determinants of growth for an insect vector population. We saw last time how the Antelope Island study was analyzed really quite simply using graphs and tables and such like, and we got managed to come to the conclusion that yes, odor-baited traps and targets could be used to control and indeed eradicate closed populations of tsetse flies. But what we then found was that there's more interesting stuff in this data, because we've got these really nice estimates of the way in which the population is changing, the way in which mortality is changing. And the question is, could we actually build a model which actually explains all of that in terms of observed climatic conditions, observed imposed trapping, and biological effects on the population? Why would we want to do that? Well, it could potentially allow us to predict the natural range of tsetse and how this might be affected by changes in climate and other environmental factors. And this is particularly interesting in these days of undoubted global climate change. Who's going to be interested? Well, people, of course, in the tsetse world will be interested, but more generally, for those who are interested in vector-borne diseases such as malaria, those interested in the effects of climate change on all vector distributions, more generally those interested in modeling insect populations, and even those interested in modeling any biological population. So it's it's a nice data set. And can this be narrowed down to a question about specific quantitative relationships? Well, in our case, absolutely yes. What we'd like to know is what effect do temperature and other climatic effects have on the growth of tsetse populations. What is it that makes a tsetse population tick? So before we get stuck into this, we want to make sure that we're not reinventing the wheel, and let's find out, has anybody else done this kind of stuff before? Well, yes, even way back in the 1920s, workers noted there was a correlation between the number of tsetse flies that you had and the saturation deficit. And later on, modeling studies were done where simple simulations were done to show how the numbers of flies changed caught on fly rounds. So that sort of simple modeling has been done. Are there shortcomings in these studies? Yes, absolutely. People didn't really take account of what the temperature variations and how this impacted on tsetse flies. And they would generally, when they were doing the simulations, they were working with catch data rather than estimates of the absolute population numbers. And also they were using rather long 5-day time steps. So what we would like to do is to find useful parameter estimates or data sets that we can actually use in doing this modeling. Now, the data that we're going to get comes from the actual the Antelope Island experiment, and quite a lot of the parameter estimates come from there as well, but also from 50 years of work that have been done on tsetse flies in the laboratory and also in the field. And what we find is that temperature is really centrally important to an awful lot of things. For instance, here we see the pupal duration declines exponentially as temperatures increase, going from 20 days at 30 degrees centigrade to 100 days at 16 degrees centigrade. Also, the length of time between the production of pupae also declines exponentially. So basically between 8 and 10 sort of days, or maybe 7 and 10 days over this range of temperature. Also, when the temperatures go up, the mortality goes up in adult flies, and in fact, it also has an effect on abortion rates. The rate of abortions go up as the temperature goes up. So all of these things, temperature is centrally important. So here are the data that we've got. We recall from part 1, the lovely data set. Nobody's got data like this really on where we've actually got estimates of the absolute numbers week by week over a 4-year period. And what we'd like to be able to do is to explain the mechanisms which are involved in this population going up over here and going down and ultimately coming down and declining to zero. Where do we start, and what kind of a model should we use? Well, we've got more than one option. And you've heard about various, when you in this course, you've heard about, and you will be hearing about different kinds of models. They're deterministic or stochastic, and they can treat individuals as discrete individuals or continuous treatment. And we could, for instance, do modeling. People have done modeling with antelope data using differential equations. So we've got a continuous time deterministic approach. We're not going to do that. We're not going to spend time on that. We're going to look at a stochastic type of thing where we're looking at discrete time. And let's have a look how that happens. And we've got more than one way of looking at that. We could, for instance, use an agent-based model or an individual-based model where we actually look at the way the way in which individual flies go from day to day. And each day we look at that agent and say, did that agent survive that day? And if it's a female, if it survived that day, did it produce an offspring? And you keep following those through until every single fly has been looked at through the entire population over the 4 years. But of course, with a standing population of tsetse flies that peaked at about 20,000, this is going to require an awful lot of computer time. So we actually went for a second option because we didn't want to use that amount of time, where we do use a daily time step, but instead of following individual flies, we follow the fate of a cohort of all pupae deposited on a given day, and we just follow that cohort using a deterministic process for calculating day-to-day survival. So this is the kind of thing that it looks like. We start with pupae, and a pupa hatches, and it either gives rise to a female or a male. Males not terribly interesting, I'm afraid. We're not terribly interesting compared to the females. We don't produce anything. Of course, we mate with the females, but it's the females that are doing the producing. So here comes the female, and we look at each day whether she actually survives from day to day, and if she survives to something like 16 days, she produces her first offspring, and that now becomes a pupa and comes back into this thing over here. And then the female keeps on going, and she goes on for another 9 or 10 days, let's say, and then she produces a second pupa. And then she keeps going until such time as she dies. And we follow these, each of these females right the way through, noting the number surviving to produce pupae on days I1, I2, etc., etc. And what we do is we actually, the progression through these things from through the pupal stage like this and through the adult stages are all temperature dependent. We know how that changes. We've seen this already. And the survival from day to day is also going to depend on various parameters, such as the temperature on that day, the humidity on that day, the trapping intensity on that day, and also possibly on density dependent factors. So how do we start this off? Well, we know what happened. Vale put 4,000 pupae on the island. So we've got this initial population of pupae, and they're distributed across a certain age range. And we just look at those ages, and we follow each one of those cohorts. And we simulate day by day the pupal survival to adulthood. And then we simulate the survival through the interlobal period of the females. Of course, the males we look at as well, but the females are more interesting. And we accumulate the pupae born at the end of a pupal at the interlobal period. And we do that for each, all of the pupae until the number of pupae in a particular cohort is actually declined. So there's fewer than one left. And then we say, right, that cohort is extinct. And we now hop to the next day and we carry on. And of course, as we're doing this, we're producing pupae, which we'll be picking up later on down in the experiment. And we just keep doing this right the way through for every day of the experiment over the 4 years. And we look at the males and the females. Okay. So that's actually the nuts and bolts of actually looking at it. What we then do is we will actually be generating the numbers of adult flies in the population on any particular day in that 4-year period. And that total number we will regard as the dependent variable, the number of adult flies. And that's our, in our model, that will be the predicted numbers. But of course, we've already know the estimated numbers from the mark-recapture experiment. And the difference between that gives us a loss function. And we can look at the sum of the squared differences between the observed and the predicted estimates, which gives you the residual sums of squares with which you're all familiar from statistics. And we look at that loss function. We weight each one of the observed values by the inverse of its variance. And so we looked at a weighted sums of squares. Now, all of the development rates, as I said before, are defined as fixed functions of temperature, and we've looked at that. So what we'll do is we'll start off with an extremely simple model. It's the one that we know it's not going to work terribly well, but we just to kick the thing off. So we assume that there's no pupil death, for instance, and that the male death is independent of all environmental factors. And the only thing that we put in there is let's suppose that female mortality increases with temperature. Do we expect a good fit with such a model? Well, obviously we don't. And indeed, that expectation is realized. So here are the numbers of male flies right the way through the 4-year period. And if we allow we basically use a fitting routine, which just basically jiggles the value of the coefficient of the effective temperature on female adult mortality to go up and down. And what the fitting routine says, well, if that's the model you've got, I'm sorry, but this is the best I can do. I can't actually get any of a better fit to the data than this. So as expected, our model gives a very poor fit to the data. Having got that one, what we do now is to say, all right, let's add another parameter in there. So let's now allow as well that male survival is also a function of temperature. So now we've got male and female as a function of temperature, and we run the optimization again. And then what we see is, well, we've got a certain residual sums of squares from the very poor fit. If we add in the effect of temperature on male survival, can we do better? And we use we do a statistical test to see whether there's been a significant reduction in the sums of squares. And we either accept or reject the added parameter into the model. And we keep doing this, keep on adding models, and each time, hopefully, we actually reduce the residual sums of squares. So we put in climate variables like the temperature, effects on the pupae, effects on the adults, humidity, residual saturation deficit, and also how many traps we were using on the island, whether there are any density-dependent effects. So we put in all of these factors, and we just continue this until we can't actually find any more parameters that we can add which are going to give a reduction in the sums of squares. That doesn't mean we've got the best model. It means only that we can't actually find anyone which is better, which is a different thing, of course. And when we do this, what we actually find is that we have got a model which basically says, well, the pupil survival, this is 5, is a pupil survival, which is e to the minus, and this is the mortality here. Well, you've got a constant term, and you've got an effect of the temperature on the day multiplied by some coefficient. So that's all that's happening with the pupae. And with the females and the males, basically what it's saying is, yes, you've got a constant term, and you've got an effective temperature, and then you've got an effect actually of the time of the year, regardless of the temperature, and finally you've got an effect of the how much trapping you're actually doing, the trapping load that you put in. And all of these, what you do is the model basically, the fitting routine essentially changes all of these parameters, all of these coefficients of the parameters up and down until such time as it gets the minimum sums of squares. And what you get actually, given that these are biological data, and we know that perhaps there's a better model out there, but actually we can get a seriously good fit to these data right the way across there. So we've actually done it. We've actually got to the end of part 2. We've actually managed to come up with the way in which we've basically shown that temperature is by far the most important parameter in the system. We can what I did was we tried actually putting in relative humidity and saturation deficit, cloud cover, and all those kinds of things. And basically the model says no. Once you've got temperature in there, that basically decides it's just a question of what the temperature is, what time of the year it is, and also how much trapping you've done. If you want to read about this in more detail, this is the paper in which it is described. Have another break, and then we'll finish up with part 3. Thank you.